I'm Yueming Dersjant-Li, Senior Scientist with DuPont Nutrition and Bioscience. I will present abstract number 149 entitled Effect of Novel Consensus Bacterial 6 Phytase on ileal amino acids digestibility in young broilers. This study is in cooperation with Massey University. First, I want to refresh your memory on what is phytate and phytase. Phytic acid consists of an inositol plus 6 phosphate groups. It's called IP6. Phytate is the salt of phytic acid. Phytase can stepwise cut off phosphate groups. When one group is removed, it's called IP5. Another group is removed, it's called IP4, and so on. IP6 and IP5 can have a strong binding to calcium, amino acids, and trace minerals. The lower esters, IP4 and IP3, have low binding capacity to calcium, but still can have a negative impact on endogenous proteins and trace minerals. The negative effect of phytate is well recognized. It can bind to protein, amino acids in the acidic environment, upper GIT, reduce the digestion of amino acids, and it can also bind amino acids in the small intestine via minerals. Phytase has two functions. One is the phosphoric effect. It can break down phosphate group from inositol, increase the availability of phosphorus. This can take place throughout the GIT. The second function is the extra-phosphoric effect. Phytase can break down the complex of amino acids in phytate, or it can break down phytate and to avoid the formation of amino acids in phytate complex. It can reduce other negative impact of phytate on digestive system, therefore improve digestible amino acids and energy. An effective phytase needs to be active in the upper GIT to break down phytate quickly and completely, therefore reduce its anti-nutritional effect. To do so, I want to introduce a new phytase that is developed by DuPont and recently marketed. It is a novel consensus bacterial 6 phytase variant with treat name Axtra PHY GOLD. I will call it PhyG in the next slides. This phytase is unique because it is highly active at low pH and wider pH range. It has almost 2 times of the relative activity in the pH range of 3 to 5 compared to the activity at pH 5.5. Therefore, it can be effective to reduce the binding of phytate to amino acids. It can break down IP6 and its lower esters quickly and completely, as shown by the right-hand graph. It is expected such a phytase may have a higher extra-phosphoric effect. The objective of this study is to determine the efficacy of this PhyG on apparent ileal digestibility of amino acids, phosphorus retention, and bone ash. 4 treatments were tested, including a positive control, adequate in nutrients, a negative control with reduction of energy, amino acids, calcium, digestible phosphorus, and sodium. We applied also sodium reduction because phytase can improve sodium digestibility as well. The 2 test dyes are PhyG 500 and PhyG 1000, that is the phytase added at 500 and 1000 FTU to the negative control. 8 replications were used for each treatment with those 3 or 8 male broilers. They were fed common diets for 5 days and followed by test diet from 6 to 15 days of age in mesh form at lab. Excreta samples were collected for last 4 days for phosphorus retention measurement at day 15. Distal ileal digester samples were collected for AID of amino acids, dry matter, nitrogen, and growth energy. Tibia edge was measured from a pooled sample of 6 birds on day 15. Dyes are based on corn soybean meal with small amount of rapeseed meal and rice bran. MCP is removed from the negative control. Negative control is formulated with reduction of energy, protein, amino acids, calcium, phosphorus, and sodium. The analyzed phytate P content is 0.33%. We used a mean particle size limestone with 380 micrometer with solubility at 45% at 5 minutes at pH 3. The analyzed phytase activity was 541 and 991 FTU per kilogram above negative control. It's very in line with targeted phytase dose. The apparent ileal digestibility coefficient is calculated based on indirect marker method. Digestible amino acid content in the diet is based on AID multiplied by amino acid content in the diet. One way ANOVA was used as the statistical difference. The mean separation was done by Turkey test or contrast test, significant level at P below 0.05. First, let's look at phosphoric effect. Use phosphorus retention as response parameter. PhyG at both 500 FTU and 1000 FTU improved phosphorus retention compared to negative control, and we do see a stepwise increase with increasing phytase dose. Tibia edge showed similar response. PhyG at 500 and 1000 FTU stepwise increased tibia edge compared to the negative control. PhyG at 1000 FTU compensated 0.22% digestible P reduction and maintained tibia edge compared to the positive control. Next, let's look at the extra-phosphoric effect. AID amino acid coefficient is presented in this table. PhyG at 1000 FTU showed a significant improvement on all amino acids digestibility compared to the negative control. PhyG at 500 FTU showed either numerical or significant improvement on amino acids digestibility compared to the negative control. We calculated the digestible amino acids content based on the AID digestibility coefficient and amino acids content in the diet. With this, I calculated digestible amino acids percentage improvement above negative control at phytase dosed at 500 FTU. At this dose level, phytase improved the digestible amino acids in a range of 3.2 for methionine and 11.3 for cysteine, with average of 5.9%. Using the similar calculation, at 1000 FTU, the improvement on digestible amino acids above negative control is greater than 500 FTU. It is in a range of 3.7% for methionine and 14.2% for cysteine, with average of 7.5%. To simplify the analysis, I use the contrast test to compare phytase treatment with negative control for AID dry matter, nitrogen, growth energy. For these parameters, we observed a significant improvement in phytase treatment compared to the negative control. Inconsistent results has been observed from literature on the effect of phytase on amino acids digestibility, which may be related to the characteristics of phytase. For example, if the phytase is active at low pH. To compare to the literature with current study, I cited a review study from Cowison and co-authors from 2017. These authors collected the data from 24 peer-reviewed papers with 745 observations published between 1996 to 2015 in the dose range of 500 to 2000 FTU. Based on the meta-analysis of peer-reviewed study, the authors concluded that the mean AID amino acids increased by 4.1%. This analysis is based on the coefficient of amino acids digestibility. To make the data comparable with this review paper, I have calculated also the percentage improvement based on coefficient. Therefore, the values is slightly different from the values presented in the previous slide. With this calculation, we observed PhyG at 500 FTU on average improved amino acids digestibility by 6%. PhyG at 1000 FTU improved amino acids digestibility by 7.8% on average. Clearly, the data from this study demonstrated that this phytase is highly effective in improving amino acids digestibility, demonstrating a higher extra-phosphoric effect. In conclusion, this consensus bacterial 6-phytase variant is effective in both phosphoric and extra-phosphoric effect. With this, I want to thank you for your attention. If you have any questions, please feel free to contact us. Thank you.